before you guys get to see all of your exciting mutations that you may or may not have made, I'm going to talk to you guys a little bit about how we do this in a cutting edge way in the lab. So you guys just made a mutation, or, well, some of you guys made a mutation. There's a lot of stuff in that, right? There are a lot of pieces to do that. And a lot of those things we can't do in a way that would help you and me. We can't do a PCR inside our cells. We can't add primers to our cells that have mutations to copy your DNA. We can't get rid of all of the original DNA, right? Like, that's not going to be good for you. So the way that we make mutation in the lab here, it's very functional. It is something that I do in the lab all the time, but it's not a way that we could actually use to cure a human disease based on a mutation. So scientists have been looking for ways to do this since we've really known about DNA. And so I'm going to talk to you today about one of those methods that is kind of the up-and-coming, newest of the new, called CRISPR. Who has heard of CRISPR before? Who feels like they know how CRISPR works? Okay, great, perfect. Okay, so we're going to start off. I'm going to give away why you should use UV lights on your eyes. They cause mutations. You guys are constantly being bombarded by things that cause mutations all the time. You have DNA that has probably been mutated from the time that you were born. Like that's just, it is what it is. However, there's a lot of DNA, and a lot of that DNA belongs to cells that are going to die off anyways, like your skin cells. So when you get radiation from something like a UV ray from the sun, a lot of times that's not a problem because that cell ends up dying and not dividing into new cells, and that's okay. It can become a problem if you guys have heard of melanoma. You can have mutations that cause problems like cancer. But the vast, vast, vast majority of mutations that your DNA undergo on a regular basis don't do anything. And a lot of that is because they're random. So anything, if you guys have ever had an X-ray, they put something on top of you, usually some sort of lead blanket. That's to block your DNA from being mutated. UV rays, asbestos, cigarettes, all these things cause mutations kind of randomly throughout your genome. A lot of times that makes absolutely no difference. Sometimes it can be bad. Hypothetically, sometimes it could be good. But in general, this is uncontrollable by us. I can't put you out in the sun and say, I want to mutate gene X from an ATG to a CTG. Like, that's not going to happen. So in biology, we want to be able to do that. We want to be able to make these targeted mutations. Can anybody think of a reason why you want to be able to make a specific mutation? Yeah. Yeah, there's a lot of genetic diseases that we know exactly what gene is responsible for them. We know exactly what mutation causes them. If we could switch that to something that doesn't cause a disease, that'd be great, right? That'd be fantastic. Now, there are some other reasons why other people might want to make this, and we'll talk about that at the end. But in general, the biggest reason we want to be able to make specific mutations is for curing diseases. So you'd like to be able to turn this T into a C, and maybe that makes a big difference. So this is a method, CRISPR. I'm going to show you guys a little video, and then we'll talk about it. This is kind of the molecular biology behind CRISPR. CRISPR is a method, the newest method that people have come up with to try to make these mutations specifically in DNA. This is just a quick background.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered, regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
So, just so you guys know what's happening, in your nucleus, this double-stranded piece, that's your DNA. That's yours that belongs to you. The way CRISPR works is it has this protein that comes in and it sits on your DNA and it kind of opens that DNA up. So this RNA piece can kind of guide through those DNA and it looks for this sequence that it matches. These are usually about 20 nucleotides long, about the same length as a primer is. So it can recognize unique sequences in your DNA so that it only cuts at a specific point in your DNA. Cutting DNA in general is bad. Bad, 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 bad. But if you can cut it at the specific point that you want, and then you can add a piece back in, or you can change a specific nucleotide, then you can make these very specific mutations. And that is how all of these genome editing technologies work. It's just that CRISPR, the reason why it's so big, is it's the easiest. It's very easy to manipulate. It's very easy to use in the lab. I use it in the lab all the time. There are other ways to do this. They're hard and they're expensive. So that is why CRISPR is the new thing. It's not that it does anything brand new that we didn't know about before. It's that it does it easier and cheaper than before. So, all right, so just a little terminology for you guys. You have a nuclease. Nuclease is that protein that cuts the DNA. So CRISPR is a multi-piece system. This is not in our bodies, so we have to put it in there. So we have to put in a nuclease, which is, again, something that cuts your DNA. The guide RNA, this is something that we create in the lab with a specific sequence that tells that nuclease where to cut. So one of these or the other of these, they don't really do anything on their own. You need them both in order to cut specifically where you want. And then lastly, a lot of times we're putting in a donor or a repair piece of DNA. So something that can take out that piece that we didn't like and that will put in new base pairs that we do want. And that's how we make these specific mutations rather than using like UV rays where you can make a mutation but we don't really know what's happening there. So 3 pieces, at least 2, sometimes 3 pieces for CRISPR to work. And this right now, I believe, is in clinical trials, or at least I know that there are trials going on with primates, so monkeys. I don't know if we're necessarily in humans yet. When we get there, there are a couple ways that this could be used. So there are 2 ways that you can do gene therapy is what it's called, right? That's changing the genes in your body for a therapeutic reason. One is called somatic cells. Somatic cells are all the cells in your body that aren't going to turn into another human being at some point. So not eggs and not sperm. Everything else in your cell is a somatic cell. It may divide, it may turn into an organ, it could be a stem cell, but it's not going to become a baby at some point. So this is where a lot of this kind of technology is hoping to be used. So in people who already have a disease, could we go in and change all of those cells to fix that gene without then passing that on to your children? The other option is called Germ Line. Germ Line is basically something that you would pass on to your children. So did anybody hear, maybe a month ago now, there's a scientist in China who said that he used CRISPR on babies. This is something that, we'll talk about the ethics of it, but this is what he did. He used CRISPR in an embryo that was in a petri dish, so basically artificial insemination. So he used CRISPR and tried to correct a gene mutation. He didn't try to correct a mutation, he tried to give these children a protection against HIV. There's a lot of reasons why this might not have been a great idea, but in general, one of those is ethical concerns. There aren't a lot of guidelines right now for how or why you'd want to do that. And in general, it's hard to do the research for this. So if Germ Line gene therapy happens, it's probably going to be a long way down the road, yeah.
Wait, when you were saying, like, for somatic cell therapy, you wouldn't pass it down to the offspring, would that mean, like, you wouldn't pass the new genetics down, or you wouldn't pass the old one that was, like, had a mutation?
You wouldn't pass the new one down.
Okay.
So specifically the way that we would like CRISPR to work is if, and I'll show you an example of a couple of these diseases here. So like sickle cell anemia is a disease just of your red blood cells. So the idea would be that you would mutate the genes that are causing just this disease in your blood cell making cells. And so instead of having cells that have this kind of sickle cell look and get stuck in your blood vessels, you would be able to mutate just those cells, put them back in your body, and then fix that problem, yeah.
So you have to make a special guide RNA for each separate mutation, but you need a specialized protein for each different mutation too, or can it be one protein that can go at all?
It's that same protein, that nuclease. You could give it 20 different guides, and it'll cut in 20 different places. So it doesn't care what guide RNA it's getting, yeah. Which is one of the things that makes this such an easy system to use, is that a lot of the pieces stay the same, and the RNA is easy to make. So potential benefits for things, diseases that people are already trying to solve. One of them, sickle cell anemia. A lot of these are maybe diseases you've heard of. Cystic fibrosis. This is a disease where basically your lungs get filled with fluid, and you have a really hard time breathing because you're just kind of coughing up a lot of this kind of mucousy stuff all the time. This is a really huge problem for children who get it. A lot of them have a very decreased life expectancy. It's very hard for them to act like normal kids. And there is one specific mutation in one specific protein that causes it. So if we could change that specific protein, you could cure this specific disease. And later in life, Huntington's disease is a horrible deteriorative disease where you get into your 30s, 40s, 50s and start losing motor control, so function of your arms, your legs, your ability to do things. And it's caused by, again, a mutation in a single gene that ends up causing kind of holes in your brain tissue. So there's some really, really kind of drastic diseases that are not super uncommon that we could fix with this technology. So getting this working there's a huge push for this. So it sounds great, right? Solving diseases, we're being specific about it. The problem is we're not quite there yet. So the biggest concern that people have right now is called off-target effects. Yeah.
So with somatic cell, then there's it's not passed on, it's just for that one individual. And so if you cure that one individual, their child could still have to deal with that? They
could still pass that gene on, yes. Hypothetically, you could then do the same sort of thing. Curing-wise, at some point we'll probably get to the point where you would want to cure those in the germline, but especially for females where you have all of your eggs before you are born, trying to stop those things before they start can be a little bit of a chicken and an egg question.
So it's a treatment versus a cure then?
It would cure the disease, but it would not cure you from passing it on to your offspring. So it would cure it for you, and then hypothetically your offspring could be cured as well. It just wouldn't go down through the generations.